Thank you. It's great to be here. I'll be giving you a background on 4D Molecular Therapeutics. We're an AAV gene therapy company, and we're focused on unlocking the full potential of gene therapy through the creation and development of novel AAV vectors, which allow us to generate best-in-class AAV gene therapeutics. So how do we achieve these goals? We're really focused on four key value driver areas. The first is our platform of vector discovery. This is termed therapeutic vector evolution. This allows us to generate optimized and highly proprietary AAV vectors for use against any organ or any important tissue in the body. Secondly, we have a very robust patent portfolio generated through this composition of matter that we discover with therapeutic vector evolution. Given the desperate need for novel vectors to get beyond the low-hanging fruit disease indications that other companies are going after today, there's a huge desire for improved vectors across a broad range of tissues in the body, and because of that, we've been able to generate 5 partnerships to date, including partnerships with Pfizer in the cardiac area, Roche in the area of the retina, Unicure for liver and CNS, AGTC in the retina, and Benetech. Finally, we have a robust and very broad product pipeline, which is supported by our own products. Our own lead products currently are for choroideremia using an intravitreal injection rather than subretinal, which is used with the current vectors, and cystic fibrosis product. And then we have a deep and broad pipeline with our partners as well. So this is the overview. Again, we've been operational for about 2 years. My co-founder is David Schaefer from the Department of Bioengineering and Chemical Engineering at UC Berkeley. Again, therapeutic vector evolution allows us to develop customized best-in-class therapeutics that are really optimized for human use. These are highly targeted to specific tissues or even cell types within tissues in the body. They're also designed to be administered to patients using routine and optimal clinical routes of administration. And finally, we can build in antibody evasion technology to evade preexisting antibodies, which really limit the use of many of the primitive AAVs in products to date. We have an AAV library of on the order of 100 million novel variants generated at 4D. And this gives us a pool of AAV vectors to select from that's on the order of 10 million times greater than other companies in the field. As I said, the composition of matter that comes out of these selection processes is very strong, and this has also led to a number of very important partnerships, not only with companies but also nonprofit organizations. We have investments and grants from organizations including the Cystic Fibrosis Foundation and the Choroideremia Research Foundation. Finally, given the breadth of this technology, it allows us to go after a wide variety of tissues in the body and disease indications, which gives us a very broad and deep product pipeline. And we're based in the San Francisco, Berkeley Bay Area. So just briefly, what do these vectors look like after we create them? Again, we're trying to create vectors that have the features that patients need for effective therapies. The first order of business is to generate vectors that target specific cells and tissues in the body and de-target other tissues, giving us better efficiency of delivery and safety. Secondly is we evolve these to use routine routes of administration. So for the retinal targeting, for example, we use intravitreal administration rather than a subretinal surgery. For skeletal muscle and heart, we are developing vectors for IV delivery. For the brain, we use intrathecal delivery rather than the intraparenchymal transcranial injections. And finally, as I said, we have the ability to expose the vector pool to human IV immunoglobulin to develop vectors which evade pre-existing immunity in the population, which limits the patients that you can treat and also limits the efficacy of the vectors and products in patients with pre-existing antibodies. So finally, just to give you a sense of the breadth of what we're doing, we have 15 discovery programs ongoing. Roughly half of these have already generated lead vectors, which are looking promising and are highly proprietary. Just to give you a few examples, we have a number of programs in the retina with intravitreal administration. We have a lead lung vector that targets lung airway after aerosolized delivery. We have IV vectors for targeting the heart, skeletal muscle, and liver, as well as synovial joints. And as I said, we have intrathecal vectors targeting the brain and dorsal root ganglia. So I think, as you'll see here, we have a very broad, robust discovery program, which we think will lead to the best vectors and hopefully the best products for patients in the field. Thank you.
Great overview. I guess first, can you tell us a little bit about your selection process? It's a big feature of 4D. What makes it unique?
Sure. So therapeutic vector evolution really is a discovery platform which applies the power of evolution to select what you want. So just like any biological evolution, we start by generating massive diversity. So we take the 10 existing serotypes that are currently being used in the field, and we create massive diversity around those using a variety of proprietary technologies. So anything from specific mutagenesis strategies to peptide insertion strategies with random 7-mer peptides, chimeric shuffling, and then combinations thereof. We then apply selective pressure to that pool of AAV vectors to identify a handful of vectors that will have the features that we want. So for example, if we want to go with an intravitreal injection and target the retina, we administer the entire 100 million vector library into the retina of a non-human primate. I should say this is all done in non-human primates or in primary human tissue, not in lower species. And then we harvest the retina after several weeks, take it back to the lab, break it open, or break open the specific cells even, or the layer that we want to target in the retina, grow those up, re-administer those AAV vectors to the retina, to the intravitreal space of another non-human primate. We do that 4 to 6 times, and we'll get a handful of maybe 10 to 20 very, very good vectors that are able to target the specific tissue or even the specific cells in the specific tissue that we want to target.
Great. And you mentioned a couple different partnerships. I think you guys have signed 5 different collaborations in less than 2 years. What makes 4D kind of a partner of choice? And can you just tell us about some of the more recent deals, like the Pfizer deal?
Sure. I think it's just a reflection of the power of therapeutic vector evolution that Dave Schaefer pioneered at UC Berkeley about over the last 15 years or so. That expertise, the libraries that we've generated, and the technical know-how, and really the desperate need for improved vectors for the field drives the partnering interest. Our most recent partnership was with Pfizer. It's around several targets in the heart. Roche, we have several targets in the retina. And with Unicure, we have liver and CNS. AGTC is a retinal partnership, and Benetech is focused on retina as well. What we try to do when we set these partnerships is give our partners exactly what they need in terms of exclusivity for a specific vector and a backup, plus a specific transgene payload for the disease they're going after. But we retain the rights and the ownership of all vectors in our library, and retain the rights to use those same vectors with different transgene payloads for the same or different diseases. So it allows us to give our partners what they need, but also retain rights to vectors for developing our own products, which is really where our value is going to be created in the long run.
So I think I saw 15 different programs on your proprietary slide. It's a lot going on. How do you think about prioritizing and optimizing other factors beyond just the vectors that you've talked about?
Yeah, well, we felt that we had a window of opportunity to really lock up the best vectors for the most important tissues and organs in the body, because obviously other companies are going to come looking for optimized vectors as well. So we wanted to really lock up the IP on the best vectors for a range of organs in the first couple of years of our existence, and we're well on the way to achieving that. So that was really job one. Job two is really bringing our own internal products forward to the clinic. Our first product will be for choroideremia. This is with grant and financing support from the Choroideremia Research Foundation. This is using intravitreal injection, which is a major advantage over subretinal approaches. We expect to file that IND in the second half of 2017, and then right behind that is a product for cystic fibrosis using aerosolized delivery of a lung-specific vector.
Great. And is there anything else we should be kind of looking out for in the meantime before you file those INDs?
Well, I think the big value drivers for 4D this year will be characterization of these vectors. As you've seen, we've made a lot of progress on the discovery of novel vectors and filing provisional IP around those. But the next big step will be to validate these vectors head-to-head versus the currently available primitive vectors and show that these are superior. We'll be doing that through marker gene studies in non-human primates and in primary human cells over the course of 2016.
Great.